Ines is a postdoctoral researcher at the Laboratory of Plant Biotechnology and Biomolecules at the University of Tours. She started her scientific journey back in 2008 as a PhD student in Porto University, with the purpose to promote the elucidation of the alkaloid metabolism in Catharanthus roseus by analyzing and characterizing the idioblast cells at 3 omics levels: metabolomic, proteomic, and transcriptomic. Once she finished her PhD, she worked at the Institute of Biology Leiden in the Netherlands, where she focused on the modification of transcription factors activity to stimulate alkaloid production in Catharanthus roseus suspension cells. She joined the lab BBV in 2015 with the desire to explore the potential of engineering the TIA pathway. At first, she mainly focused on the identification of missing enzymes of the anti-cancer alkaloids pathway by combining genes. She studied its correlation by functional assays and transient gene inactivation, VIGs. These results were able to shed light on the biosynthetic order governing the synthesis of major roots alkaloids, leading to the production of non-natural alkaloids in yeast by metabolic engineering. These results laid the groundwork for the metabolic engineering of tabersonin lochnericin derivatives of pharmaceutical interests. The impact of these results led the team to participate as partners in an international European consortium, Miami, in which they use Rauwolfia serpentina alkaloids as a case study to implement an efficient flowchart approach for pathway discovery of novel natural products in order to allow cost-efficient and sustainable production of rauwolfin, serpentine, and alstonin in sufficient quantities to allow bioactivity testing. Ines, I let you speak now. Ines, you have to turn on your micro.
It's okay now? Yes?
Yes.
So thank you so much for this nice presentation. A lot of words just to say that I'm a big fan of alkaloids. So thank you so much, the audience, to be here today to listen, this opportunity to listen one part of our work, and the host members for this possibility to widespread a technology that is crucial in our lab to implement our main goal, that is to unravel the metabolism pathway of pharmaceutical compounds, alkaloids. So today, we're going to talk about a specific technique, virus-induced gene silencing in cirrhosis. Basically, this, I'm going to share my presentation. It's okay?
Yes, it's not full screen. It's full screen
yes.
now. Yeah.
Okay. So basically, I will make a brief introduction of the technique, the hands-on approach, how we apply the technique, and the outcome, how using this technique was crucial to identify some candidate genes. So in the past, not so long time ago, when we wanted to study a gene, basically we had to wait to observe a mutation, then study the mutated gene. For that, we needed to wait or the nature to be kind enough, or we forced nature by promoting mutagenesis. a technique that looks like primitive, but it's not that ancient. And not long time ago, it was widely used to study, for instance, dwarf genes in Arabidopsis and reveal a lot of important information that today we use. However, in the present times, we need more robust techniques, especially because we evolve so much, we develop so much techniques that allow us to identify so many genes that we needed to be more sharp in the way we approach the characterization of these candidates. So all the previous methods had something in common. They were time-consuming, and our time is priceless. Especially in today times, where we developed the RNA-seq, where we are able to identify genes on the spot where reactions happen. So if you have several candidates, it was urgent for us to identify, to develop a technology that easily and fast we could screen the valid gene. That's when it comes the transiently silencing gene through virus-induced gene silencing. That basically is based on the mechanism of defense of the plant, RNA mediated toward many viruses, which targets basically viral RNAs or transgene RNA products and promotes their degradation in a system that I'm pretty sure that you remember from the classes of biology, the RNA-E interference. Basically, the process, the double-strand RNA of virus, is recognized by the machinery of the plant, by the RNase decer, cuts this double-strand RNA, forming the interference RNA, small strands RNA, that are recognized by the RISC protein complex, that by itself it will search the complement chain of our RNA that will block the expression of viral proteins by impairing their translation or promoting the degradation of the RNA, and like this, impairing the proliferation of the virus. The VIGs use this system in their profit to silence the desired genes. There are several articles that you can search to better understand the mechanism that is behind this technique. If I can advise you, some publications recently, two leading members of our lab, they publish a very nice resume that also tackles all the most recent publications. So it's very easy to understand, to read and to understand the technique through this article. And also they were the leading editors of a publication that gathers a wide spread of technologies and methodologies that are able to using this technique to apply in different plant species. It's a very useful hands-on approach book. Basically, the VIGs, we identify a candidate gene, we amplify a portion of this gene, a fragment with no more than 500 base pairs. We clone in the virus vectors that we already identified that are capable to infect our plant. We inoculate our plant with these vectors harboring a small portion of our candidate gene. The vectors are able to multiply inside of the cells, spreading in the cells, through the plasmoderms between the cells, and through the organs of the plant, through the phloema. Inside the cells, these double-strand RNAs that harbor our fragment gene will be recognized by the decer, will be cut in the small interference RNAs. These small interfering RNAs, namely our fragment, it will be recognized by the RISC. The RISC will bind with the complement mRNA, which is going to be our own gene, will recognize, will bind, will promote or the cleavage or impair the protein translation. And like this, we achieve the non-expression of our gene, which it means the knockdown of our gene. This technique presents several advantages and disadvantages. In the advantage, one of which is we can reach a high percentage of suppression of expression. In some cases, we can even reach 90%. On practice, normally between 70 and 80% in very good cases. It's a very fast method. We have a wide spread of resources, lot of vectors, as many as virus capable of infect our plant. The plasticity of the method, we can use agroinfiltration, leaf rubbing, by vacuum infiltration, biolistic, syringe, cutting, immersion. The methodology is very rich. It's very accessible. It's not expensive, unless the biolistic. We can apply this to model and non-model plants. And especially we can use this in situations where we need to avoid the knockout on essential genes. We want only to see a diminishing of the expression level. As main disadvantage, we have to, when we are studying non-model plants, sometimes we need to search the best viral vector or the inoculation method or the plant stage development. For instance, if a plant produces a lot of latex, it's more hard to perform agroinfiltration. If we want to study stage of development of the plant, where it is more developed or less developed, the method to inoculate the plant, it will differ. The silencing process also, it's many times not uniform. What this means, when we silence the expression of a gene in a leaf, we don't know if it will be in the whole leaf, half of the leaf, upper part, middle part. So when we collect that leaf, we normally collect all. The technique has evolved, and there's already some new approaches that counter-tackle this question. However, it's not that widely used. The silencing is also transient. So if we are studying a method, a mechanism, and we need time for him to be implemented, we have to have in mind that this silencing, this transient silencing, occurs for around 20-25 days, in best case 30 days. Also, when we are studying complex pathways, we might dismiss important candidate genes because the pathway tends to regulate himself. So we make knockout of a gene, and the pathway, since the compounds that accumulate tend to equilibrate, we see no effect. And we might miss an important gene. Also, it's very hard to differentiate between isoforms due to their similarity. However, this technique was crucial for the study of the metabolism pathway in our plant. So for you who are assists who are not familiar with this plant Catharanthus, why are we so devoted to this task? Well, Catharanthus roseus, or Madagascar periwinkle, it's a plant endemic from Madagascar Island that today presents a plantropic distribution. This plant, also known as the anti-cancer plant, is known by the production in their leaves of two anti-cancer alkaloids, vinblastine and vincristine. First natural compounds used in chemotherapy, and up to date, the plant is the only source of these two compounds. Also is recognized to produce more than 130 alkaloids, some of them with pharmaceutical activity, and many of them producing in such a low amounts that we are not able to perform the adequate pharmaceutical tests. So what was our main goal? To unravel the pathway for the production of these two valuable molecules, and in a similar way that it was performed with the alkaloids Taxol or with the alkaloid artemisinin to treat malaria, transfer the production to cell factories in an elegant process to make us independent of the plant. However, this task resulted to be quite challenging. The path is complex, involve a lot of enzymatic steps, involve a lot of different levels of cells, organization inside the cells and between the cells. To give you an idea, from the precursors tryptophan and mevalloprate pathway up to vanillin and cafferantin, which are the two alkaloids, backbones of the dimeric alkaloids, it happens more than 30 enzymatic steps. In 2016, 20 were already characterized. In the past, members of this group were involved or in the identification or in the characterization of some of these genes. So in 2016, we were devoted to unravel the missing steps in order to achieve the production of the monomer alkaloids in cell factories. With these two monomers, we are already capable of producing semi-synthetic alkaloids that can be used in chemotherapy. So the first thing we do, we generate an enriched gene expression network that it was really important and allow us to identify value candidate genes. But there were several. And in the traditional methods, we would have to produce the protein and then perform in vitro assays. But this would mean that we would have to know where on the pathway our candidates would be. And most of the times we didn't. We just knew by gene networking, evaluating the expression, we knew that they were involved, but we did not know where. So we needed to develop a method that easily we could see alterations in the metabolism profile, and like this confirm the activity of these enzymes or not. While in other labs, our colleagues, they always use the VIGS methodology through agroinfiltration. We knew that this would demand that we use plants already with a certain stage of development. And we know that many of these intermediate compounds, where our enzymes, our candidate genes would be involved, are accumulated in very low amounts, especially in already established plants. So we needed to develop a method to attack small plants where the metabolism is still very active, so the probability of rich intermediates that are low accumulated is higher. Since small plants of Catharanthus are recalcitrant to agroinfiltration, we had to develop another method. Since in our lab we had already experience with Biolistic, we decided to develop this method and to transfer in the same way our virus vectors to the plant through Biolistic way. So basically, and I will give you here an example that was performed by a colleague in our lab, Pamela Cruz, where by identifying a gene that at that time we did not know what was performing, we transiently transform our plantlets of Catharanthus. We wait a period of time. We collect the leaves. We perform the quantification to validate the silencing of the expression level. And then we perform the metabolomic analysis to search the involvement or not of our candidate genes. First step, choose the right vectors. In our case, and after searching and testing several, we observed that the best one was the tobacco retrovirus. Binary vectors, that one of the vectors presents the viral RNA encoding proteins responsible for the viral multiplication and dispersation between cells of the virus particles, and a second plasmid that encodes not only proteins from the coat of the virus, but also our interest gene. However, and in the book that I advise you to search in case you are interested in this technique, there's also description of several other vectors, namely the cucumber, foxtail, African, also for poplar, also vectors to be applied in trees, like olive, performed by also a colleague from our lab, Konstantinos, and also apple trees, performed by Sebastian from our lab. So there's a vast range of possibilities of virus vectors that you can or you must test before you decide which one is the best in your conditions. A second important point is that when we perform the silence, we need to have a positive control. We need to know when to collect the leaves. After 5 days, after 20 days. How we monitorize that? We collect every day and we perform qPCR. We cannot. It would demand a huge amount of plants. It's possible, but we had to engine other ways. So for that, we use positive controls. And positive controls is normally genes that once silenced give a phenotype. I present here several examples. The most widely used is the PDS, the phyto18 desatyrase gene that interferes with the carotenoids pathways and impairs the protective effect of the carotenoids on the leaves, and that is translated by bleaching of the leaves. There's also the CHS that is also widely used, but gives a more yellow effect, not so easy to identify the right moment to collect our samples. But depend on the species we are working, depends the positive control we choose. Here I want to describe you how in the lab we perform the biologic approach. So we have the gold particles. We have our plasmids that we already purified. We coat our gold particles with the plasmids. We deposit the gold-coated particles in macrocarriers, and then in specific compartments, biolistic, we deposit these particles in our plantlets. The stage and the size, the distance that this technique has to be applied, it depends between species. For instance, in the case of Catharanthus, we must see the buds of new leaves appearing, which makes sense. It's cells that are highly active in division cells, so they will accept and incorporate easily this strange genomic material. Sorry. For instance, for Hawafia, we need more developed leaves. This is also related with the capacity of the plant then after to recover from this injury. In our lab, we observed that the best method is biolistic. However, you can also apply other methods, like, for instance, agroinfiltration, by cutting and immersing in the solution with agrobacterium, by vacuum infiltration, by production of the particle virus in Bentamiana, and then by syringe extract this particle virus and introduce by syringe in your study species. So once again, the book presents several hypotheses of methodologies that you can use to apply this technique. In our particular case, once we perform the biolistic, one crucial step is also the recovery of the plants. We have to take care of them like babies. Very, very careful with them. And after more or less between 15 to 20 days, we start seeing some phenotype. Once we see the phenotype in our control plots, in our positive control plots, we are able to decide the right moment to collect our samples and then to perform the qPCR, the quantification, to confirm the silencing and the metabolic effect. And so that's how we did it. We had our candidate gene, the GO. We silence the expression of this gene in Catharanthus. We perform the qPCR. We confirm that the expression of this gene was diminished in our plant, and then we perform the alkaloids quantifications. We observe the alkaloid content. And as you can see here on your right, we saw several variations in different alkaloids, especially in two, catharantin and vanillin. Here in low gray, we can see that this diminishing is effective and pronounced. However, this is, if you remember on the pathway, this is the end product that we are interested. This still did not give us the answer where the gene would be working. In the case of the GO, we were lucky, because when we give a better look to our metabolic profile, we could see that our silenced plants accumulate one compound that we predicted to be the product of one already identified enzyme, geshoshizin synthase. That we knew it, that once to continue the pathway had to suffer an oxidation. Our candidate gene was a P450, so it was a good candidate to perform this reaction. So like this, we were able to right positionate our candidate gene. Of course, that after we had to perform the further functional characterizations, production, the protein, and in vitro do the assays, subcell localization, all the biochemistry assays. However, this avoid have to do the same with 7 candidates that would take several months to validate, just to validate. We reduce the time span in 1.5 months. Since we saw that the technique was efficient, we decided to let's go on and tackle the rest of the pathway. Using the same procedure, we were able to identify more 2 candidate genes. In this case, the genes that were involved on the final steps for the production of scatterantin and tabercenin. At this point, we were able to fulfill the pathway and, in the next step, pass through the design of the cell factory, able to produce these molecules. Also in a very elegant work that it would be very interesting for you to listen. It was performed from our colleagues in the lab. Once we saw that we could use this technique to unravel the pathway in Catharanthus, we decided to explore other plants using the same technique. We already use it and we are implementing it in other species. For now, we already have proof of evidence that we can use the same approach in Hawaphia Tetraphila or Serpentina. I hope that in the near future, other colleagues can come here and present their results in another species and how they use this technique also to validate other candidates' genes. And like this, we reach the end of this presentation where I wanted to show the potentialities of this technique. I would like to make a special thanks to the leading members that allowed the development of this work, Professor Vincent Cordavo and Professor Sébastien Bassot. Also to two previous PhD students and postdoc, Emilie and Cyrielle, to our PhD student Pamela, that she did enormous work, especially on these final works, and of course our director Nathalie Guivache, that represents all the members that direct or indirectly contribute to this work. And thank you so much to all of you for attending, and I hope you enjoy this exposition.
Thank you so much, Ines. It was a fascinating talk. I think everyone will agree in the chat. If you have questions, feel free to put it in the chat box.
Okay. Si vous voulez faire la question en français.
Oui. Or I can translate if the questions are in French. If you prefer.
C'est pas grave.
Okay, so I had myself some questions. So very interesting technique. I heard about in class, but never really saw in the field, I would say. How long did the optimization of the technique take in your model? Like you had to choose between vectors as a approach you were going to use. How long did that take, and what were the main challenges you faced during that optimization process?
First of all, in Catharanthus, I was very lucky because when I arrived, the hard work has already been done by a previous PhD student, a very good PhD student, Emilien, but he made the major work. However, it took 1 year, 1.5 years to define all the details for the amount of plasmid, the amount of particles, the distance, the time of recovery of the plants, in the shadow, not in the shadow, in which temperature. However, when it was to transfer the technique to other plants, it was much, much faster. It was in 2-3 months.
Great. I had a question about the agrobacterium technique, actually, because I'm using myself some agrobacterium agroinfiltration. And at one point you said that first you were producing the various plasmids in Nicotiana bentamina and then using them in other models?
One of the techniques, I don't use it particularly. Yeah. In the lab, we use to transform apples trees. Sébastien Bassot, Professor Sébastien, that do that. And now we have also a new postdoc, Louis Valentin. He will also implement this technique to study his plant. So I let to him to speak if one day he is his desire. However, basically you transform by a syringe Nicotiana, bentamiana with the agrobacteria harboring your plasmid. Then you promote the viral multiplication. Then you collect the leaves. You macerate in a buffer, in a solution. You collect the particles, the viral particles, and then you use that solution to infect, to inoculate your species.
Okay,
This happens.
So it's a way to amplify your viral particles.
Yes,
Okay.
this is a way to avoid the situations where species are recalcitrant to agrobacterium infection. Like
yeah,
this,
okay.
much more.
Great. I don't know if anyone has question in the chat, or you can also unmute yourself if you want to ask questions directly. In the meantime, I will have some other questions. It's okay. What would be the prerequisites for using VIGs? Do you think, in your opinion, that some models there will be too much technical difficulties to implement VIGs?
Yeah. I agree, especially trees, because of the time. Many times the issue is on the plant, not on the technique. So, for instance, the germination of the seeds, the time to the plant to achieve, to reach the right size. You want to go faster than the plant. But for instance, here in the lab, one of the postdocs can do this on olive tree. If he can do this on olive tree, I think he opens the barriers for us to explore any plant or apple tree. The good thing of the VIGs using Biolistic is that we don't think so much about if the plant will accept or not agrobacterium, because Biolistic, it's above that situation. That's why since we attack or we tackle different species, we wanted a method that easily anyone in the lab could use to study any plant. You just needed to accurate the exact moment to perform the technology. Okay. You don't need to adjust strains of agro, types of plasmids. We have a system that we wanted to apply in different species in order to make it more efficient.
Okay. I was wondering if you talked about a bit about the advantages you don't have knockout genes if you don't want them. So is there, was making comparison with CRISPR-Cas9, for example, in which you do have knockout genes. Is that the only advantage compared to CRISPR-Cas9 in your opinion, or do you see other maybe advantages?
Because
yeah.
I think that's for us the best, because if you imagine, if we do CRISPR-Cas9 on plant, and we do, if we have 14 candidates, which many times we have, if we have to do CRISPR-Cas9 and then wait for the silencing and the knockout, and then the generations, and then until we see the results, it's longer. It's with VIGs, it's 1.5 months, more or less.
So CRISPR would be more useful if you already have your candidate genes.
Oh yeah, sure.
Yeah.
Sure.
Well,
or if you do it in suspension cells, for instance.
We were wondering if anyone in the hearing this presentation was using VIGs in the lab, and if they had some insights to share. I'm not seeing much questions in the chat box yet, but your presentation was very complete and very thorough, so I guess that takes part here.
So, en français.
Yeah, mais peut-être que c'est l'anglais qui fait peur aux gens. On peut le faire en français. Oui. Je sais pas si... Enfin, moi, je me demandais vraiment quelles avaient été les grandes étapes où on se dit, OK, là, ça va jamais marcher. Ça fait 15 fois que je le fais, ça rate. Est-ce qu'il y a eu des moments comme ça que pendant l'optimisation, on se dit que ça va pas marcher? Au final, c'est quel était le déclic ou la technique
C'est les clics. Si tu pars avec Pamela, par exemple, il y a des fois que c'est la époque. La plante, elle n'est pas la même toute l'année.
Oui.
Et par exemple, que c'est beaucoup mieux si nous fait en printemps qu'en été. Par exemple.
Ouais.
Mais aussi, il y a des fois que c'est peut-être la plante et elle est d'accord avec nous en été. Même nous, c'est pas bien pour travailler, non? Nous préfèrent faire d'autres choses. Et il y a aussi l'expérience, c'est plus avec la plante qu'avec la technologie.
Okay.
La technologie, c'est un protocole. Et VIX, comme maintenant nous est implémenté dans les labos, c'est un protocole. C'est plus problématique si la plante est stressée ou pas. Si nous ajoute beaucoup d'eau ou pas, si la chambre est beaucoup d'humidité ou pas. Ça, c'est plus déterminant pour la technique qui faire la biolistique.
Et du coup, vous travaillez avec des plantes en serre, enfin en conditions contrôlées pour éviter ça?
Oui. Parce qu'après qu'il nous fait la transformation, c'est important que pour le développement de la particule des virus, la plante, elle change pour une température adéquate pour l'agrobactérium. Dans cette situation, c'est 28 degrés, par exemple. Et normalement, comme c'est un plante tropicale, elle grandit à 32, 33. Donc, nous sommes obligés à changer de température. Et aussi, comme ça...
Pardon, ça a bugué chez moi. Oui, du coup, il me semble quand même qu'on a du monde dans le chat qui utilise VIX. On se demandait aussi sur quel modèle, si ils utilisaient du coup la même technique, c'est-à-dire la biolistique, ou si pour le coup Agrobacterium ça suffisait. Après, c'est peut-être l'heure de la digestion, je ne sais pas si les gens sont... Jean-Claude qui demande quels sont les témoins indispensables à faire pour cette technique.
Ils sont là.
Les témoins indispensables à réaliser.
Les témoins.
Les témoins, les contrôles, les témoins positifs ou négatifs à réaliser.
OK. Et les contrôles, c'est les plasmides vides, forcément. Et le contrôle positif pour savoir les bons moments pour collecter la feuille. C'est l'unique contrôle. Tous les autres, après, c'est dans la plante que tu as.
Ouais. Donc le contrôle positif, ça se fait plus pendant la phase d'optimisation et une fois qu'on...
Non, tout le temps.
Ou à chaque fois? Tout le temps? D'accord, ok.
Nous jamais sait si en cette façon la plante elle grandit plus vite, donc tu vois les résultats après 15 jours, c'est la plante et ne sait pas pourquoi cette batch des plantes elle va grandir en plusieurs jours, en 20 jours. Donc nous tout le temps utilise un contrôle positif. Et l'idéal, c'est même ajouter un contrôle positif à notre plasmide. L'idéal, c'est si nous peut l'ajouter à notre gène que nous avons silencié, un gène de contrôle positif. Et comme ça, nous collecte dans la feuille uniquement la région de la feuille qui est silenciée. Mais ça, c'est une chose qui nous
est commun à d'autres
nous commence à développer ici au lab.
Oui. Je ne vois pas d'autres questions. Je pense qu'on a déjà, enfin, je ne sais pas quelle heure il est exactement, mais on a déjà tenu une petite heure avec cette présentation qui était extrêmement intéressante. Et merci pour les réponses aussi aux questions. Je vais laisser peut-être la parole à Manon pour finir cette session de webinaire qui va nous parler un petit peu du prochain webinaire. Et voilà.
Oui, donc du coup, je vais continuer en français, hein, voilà. En fait, on voulait un petit peu vous introduire le sujet du prochain webinaire, donc qui sera en juin. Donc, on va parler en fait du stress abiotique, encore une fois, avec une post-docteur, Maïté Léchevin, qui va nous parler en fait d'un de ses projets de thèse, donc sur l'étude de la réponse d'Arabidopsis thaliana durant un stress salin, par une approche un petit peu multiple, à la fois sur le métabolisme primaire et spécialisé. Donc, bien sûr, si vous voulez participer au webinaire, faire une présentation comme aujourd'hui, si vous voulez participer à l'organisation des webinaires, vous pouvez nous envoyer un email. On se fera un plaisir de vous répondre. Et donc, pour terminer, on aimerait vraiment remercier Inès pour le travail qu'elle a fait pour la présentation d'aujourd'hui. Et je suis sûre que ça en a intéressé beaucoup. Et donc, on vous souhaite une excellente journée et on se voit le mois prochain.
Merci à tout le monde d'avoir été là encore aujourd'hui.
Merci à tous et merci Inès.
Oui, encore merci Inès.